is my pleasure to introduce Marc Freeman, who is going to give the Seymour Benzer talk tonight. And actually, Marc is going to tell us about Seymour. So I'm going to leave that for him, and I'm just going to introduce Marc. Marc is currently director and senior scientist at the Volum Institute at Oregon Health and Sciences University. He got his Bachelor of Science in Biology from Eastern Connecticut State University, which I learned today is an absolute gem of a place to get a biology education, a secret in Connecticut. He did his PhD with John Carlson at Yale on Drosophila olfaction, and then moved to a postdoc with Chris Doe, University of Oregon, studying Drosophila neurogenesis. And then he began his independent career at the University of Massachusetts Medical School, where I think this is sort of where he really took off in understanding and discovering and characterizing the beautiful complexity of glia in Drosophila, amongst many other things that he will tell us about again. When he was at UMass, he was a Howard Hughes Medical Institute early career investigator, and then a regular investigator, so from 2009 to 2016, until he moved to the Volum, where presumably he's got many other things occupying him in addition to his work. So again, I'm going to let Marc tell you about his work, but I just want to briefly say why we chose him for this keynote. His work on this really important part of the nervous system is pioneering. It gave us tools to define and understand what glia there are in this model system that we really care about, understanding their function in the normal development, and also really importantly in health and disease. And he's taken his work to translation all the way to phase 1 clinical trials now. So, yeah, correct. So I think that is really something many of us aspire to. And what's really important is that his work touches on everything every one of us in this room is doing, whether we like it or not. So I think it's really important that we all think about it. Another reason that we picked Marc for this talk is that he's the kind of thoughtful and approachable and very kind person that we really value in this community. In every interaction that I've had with him, he puts people first. His mentees recently nominated him for the NINDS Landis Mentoring Award, which he won, and I just have a couple of quotes from his mentees about him. His innovative, elegant, and inspiring work was why I nominated him, and also because everyone I had spoken to had given me nothing but praises about his dedication as a mentor. And here's another one. It's undeniable that it is because he cares so much about the people that he mentors that Marc can provide them with the extraordinary training and career support. So I think that aspect of care for people is something that we really value in this community, and thank Marc for representing. So I'm going to turn over the stage to him, and really looking forward to...
Should I wear this hanger? Okay, you're going to hang me. Okay. All right. It's like winning a medal. Okay, that's good? All right, well, Avi, thank you very, very much for that really kind introduction, and you and Andy for the invitation to do this. I grew up at this meeting from the time I was in John Carlson's lab. We always came to this meeting, I think from 1994 onward. I have been a regular attendee. I haven't been for a decade, I'm ashamed to say. Partially blame that on COVID. But it's really wonderful to be back. And I, more than anywhere, I feel like, here are my people, right? It's great. So I'm going to tell you about a story that is ongoing, mainly the work of a postdoc in the lab, Yunshik Kang, with a couple of other stars, Katie Lehman, who was actually post-bac, I think. Or no, undergraduate in Avi's lab, and Rachel De La Torre, with a lot of help from others that I'll mention along the way. But first, let me start with who is Seymour Benzer? And the first comment is, shame on you for having to ask, okay? We should all know who Seymour Benzer is. He was really an inspirational person in the field. There's a whole body of work he did on recombination and phage, which was beautiful, which we don't think of him about. What we think about him is because he was the first to really make the claim that animal behaviors aren't so complex that you can't study it at a single gene level. That was one idea, is you could do screens, break individual genes, and try to understand behavior. The other in the field at the time was that can't be true. It's so complex, you're going to have to select for behaviors, and as you select more and more, you learn more about how they're controlled genetically. Both are true, but what Seymour did was develop a number of really beautiful screens for really complicated behaviors like vision-driven behaviors, locomotion, reproductive or mating behavior, learning and memory, and of course the most famous, which was recently awarded the Nobel Prize, on circadian rhythms. And so he really thought if we just break genes and look to see what went wrong and dig deep, we can understand the mechanisms beneath these behaviors. If you would like to read a book that is really a great summary of a lot of his fly work, it's this one, Time, Love, and Memory. David Stewart's not here, which is good because this is not Cold Spring Harbor Press. You can't buy this downstairs. You could get it on Amazon if you don't think they're evil. But it is really a nice summary of sort of what motivated him and what the big accomplishments were. Who are the people who you probably hear talk from in the field that did a lot of the work that moves things forward. So I also have a very personal connection, a very small personal connection. So this is the external report on my thesis when I was in John's lab that had to do with steroid hormones in the brain. So there's John, and the outside reader was Seymour Benzer, right? John knew him and managed to talk him into doing this. And I was thrilled, right? Because then I had a thesis where I had Seymour's signature in there. And I got very excited because he could have rated it anything, but he rated it distinguished, right? Now, don't clap yet. Because he had a postdoc in the lab read it who was working on how steroids might affect behavior, and he just rubber stamped what she said. I'll take it. I still hold on to that and consider it precious. So, to give a lecture named after him is actually anyone who has been in flies for 20 years or more will tell you that's really a special thing. So thank you again. Okay, now on to what we've been working on. I'm going to tell you about how we're not going to talk about a ton of different neuronal subtypes or their connectivity or anything. We're just going to look at a few neurons and try to understand how it is that they develop, they wire, sometimes they overwire, and then they get pruned back, and what are the molecular pathways that drive that. And I think what you'll see as an underlying theme for our group is that we try to define really interesting biological questions and attack them using the wealth of genetic tools that are available in the fly. And I won't have to explain a ton of those to you. The basic idea is this, that in the nervous system of complex metazoans, function is not inherent in the immature nervous system. You actually overbuild the nervous system, you make too many neurons, you make too many synaptic connections, and then there's a subsequent phase of remodeling where both the populations of neurons are trimmed and so are their connections. Now the simplest could be where a synapse just retracts and it's resorbed by its parent arbor. Or you could have things like local pruning where somehow this branch gets clipped off, degenerates, and then it gets cleared by surrounding glia. There's also whole cell elimination where cells will wire into circuits and then after a certain amount of time undergo cell death. And this was recently realized to be a very complicated process that probably requires a sophisticated conversation between neurons and glia based on the work from a number of groups. And so here, for example, is the one from the visual system in the DLGN of the mouse. Retinal ganglion cells will overwire to their postsynaptic targets. Those compete for space on the postsynaptic targets. And then over time, the strong ones win, the weak ones are eliminated. Okay, that happens. This sort of local pruning happens throughout the brain, but if you were to ask someone who works on mice or any complex system, the extent to which it happens is really not clear. It could be most neurons, it could be half, we don't know, but it is quite extensive, we believe. And then there's this other type of remodeling where neurons will be born, they'll wire into circuits, fire for even a couple of weeks, and then some of them just choose to drop out and go away. And how those are chosen and how they execute that death, we don't know. But just to give you examples, 50% of chick ciliary ganglion cells are eliminated, 40% of the GABAergic neurons and cortex in the mouse. So this is not a small thing, right? It's quite robust and quite widespread. So that refinement happens not just involving neurons, but also glia. And there is quite a bit of debate in the field as to exactly what glia do. On one end of the spectrum, they could just be garbage collectors, right? The neurons could make the decisions and then prune and leave the debris, and glia come in and just swoop it up. And then the other end of the spectrum is glia could play some active role in choosing which of these neurons, like the purple versus the green, get eliminated and sort of murder those connections, right, or whole cells. The answer is probably somewhere in between, and I'll tell you why I believe that. There's a few good examples. So in this DLGN example, where both astrocytes and microglia will move in and help shape those connections, you can do things like knock out molecules used by microglia, C1q, for example. And if they were just garbage collectors, you should still get when the circuits mature, you should get 2 connections with a lot of debris, but you don't. What you actually find is there are excess synapses. So it goes from 12 down to about 2 or 3. When you knock out C1q, you get 7 or 8, okay? Functional synapses, which implies that it's not that they're just coming along and cleaning up the mess, but there might be some signaling going on there. Now, another good example is Purkinje cells during development. They are overproduced in cerebellum. At some point, a subset of those will turn on low-level caspase-3, which is an indicator of cell death. Microglia will go over to those caspase-3 positive cells. They turn caspase-3 way up, they die, and then the microglia eat them. And if you embryologically ablate microglia, what happens is about the same number turn on caspase-3 at low levels, and they sort of keep it for a while, and then they shut it off and they live, okay? So it's examples like that, plus a lot of work from really, outstanding model organisms like C. elegans, which have shown that an engulfing cell can actually take part in pushing a cell that's half dead or thinking about dying over the edge. And I think a big question is, how much does that sort of signaling between neuron and glia take place in the nervous system? Okay. Now, another thing that struck us is this is really an understudied topic compared to things like synaptic connectivity or axon pathfinding, but a certain number of cells have been studied, and when some molecular mechanisms have been identified, they seem to be different in each place, right? So here we have in DLGN C1q, CR3, another MEGF10 MERTK, that's Draper in the fly. If you look, for example, in barrel cortex, you can get rid of CR3, and pruning in the barrel cortex is fine, right? Instead, Dory Schaefer's group showed it was fractalkine. So fractalkine is required here, but not in DLGN. CR3 is required here, but not barrel cortex. And so on, like this is a caspase-dependent death, which does not seem to take place here. So the point is just that every place we look, it seems like those cells do it differently. So there are a variety of ways in which cells choose to remodel. And the big question is, how many different ways do they do that? And if you were to look across the nervous system, how complicated would that be? So there's a lot of really big questions in neuronal remodeling that need to be answered. One is just, what are all those pathways that drive remodeling? How does a neuron choose to clip off one branch at a cell biological level, and what's the machinery that does that? How diverse are they across the nervous system? If you were able to look in an entire nervous system, would you find every cell does it differently? Or is there really some basic pruning program? If there is, we haven't found it yet, because no such program could be applied to all groups of cells. How do glia talk to the neurons during this remodeling event is obviously a question, based on the introduction, you could assume I'm pretty interested in, we're interested in. And then there's pretty good evidence, for example, in Alzheimer's disease models in the mouse, that some of these pathways are reactivated, like the C1q, CR3 pathway, and actually help promote the death of cells in that condition. So there's the possibility that some of these are inappropriately activated in disease and help drive neuronal loss in patients. So how can we address that in a system as wonderful in the fly? Well, the answer is we have this beautifully stereotyped nervous system where we can uniquely identify individual cells within segments. So here, it's 13 segmentally repeated neuromeres. Each neuromere has about 650. Someone's going to tell me that number's wrong. That's probably an old number. They know it's 642 or something. And about 65 glia. Based on all the drivers and tools we have now, they're almost all uniquely identifiable. Here's a driver labeling the corazonin neurons. There's one per hemisegment, and we can make single-cell clones, right? So we could watch these be born, watch them wire, and then we can ask, how do those change when they remodel? Now, remodeling, at least the part we're going to look at, is primarily during this first 24 hours of metamorphosis. So the animal is going to enter the pupal case, undergo about 4 days of metamorphosis and pop out as an adult. Now, I've talked about sort of activity dependence and other things. There's a second wave, almost certainly, of remodeling that happens at the ferrate adult stage and early adult stage, which I'm not really going to talk about today. You'll hear about that tomorrow from Taylor J., who's going to talk about her attempts to understand what's happening with synaptic connectivity there. I'm going to really focus on this early stage. And, of course, it's a great place to look at that because you go from a larva to a pupa within an hour, and then the entire nervous system fires off and everything starts happening at once. So you have this beautifully coordinated set of pruning events. And that's where people like Oren Schuldiner and others who have studied this have focused. And mushroom body has been one of the areas of focus. We're going to focus on the ventral nerve cord primarily. So, in those first 12 hours, for example, these corazona neurons go from beautiful neurons to completely gone. Astrocytes, which I'll show you a little bit more of in a bit, go from these cells that are associated with synapses and are helping them run operations to highly phagocytic cells that are going to devour all that debris. Okay, so those are the two main cells we'll talk about. Here is a single-cell clone of an astrocyte in the ventral nerve cord of the fly. I could tell you later why we call them astrocytes. They harbor many, many of the features of what you would want to see to call it an astrocyte in a mouse. They sit at the edge of the synaptic neuropil, this light gray region where all the axons, dendrites, all the synapses form and most of the axons and dendrite material are. This is what it looks like if you look at the entire astrocyte network. So here are those cell bodies sitting on the edge. The green are their membranes, which go in and associate closely with most, but not all synapses. And here's what a close-up of that would look like. And within just a couple of hours, so here's 6 hours after puparium formation, so after the animals enter the pupal case, you can see they've changed their morphology dramatically. So they've gone from a non-phagocytic cell to, there's about 180 of these in the entire larval brain. Every single one undergoes this transformation. So it's a really dramatic change in their morphology. As far as we can tell, every single one does. They eat a lot. Okay, so here is a marker, Bruchpilot, anti-HRP, or anti-BRP. That labels, what we would probably call all synapses in the synaptic neuropil. Within about 6 hours, that starts to go away. Within about 12 hours, most of those are gone. If you do things like just block the expression of a receptor, which we've studied quite a bit like Draper, you can see that most of that material remains. Okay, it's probably degraded. I haven't shown you whether it is or not. But that's just to convince you that these astrocytes are doing a huge amount of work. Okay, so that's astrocyte-specific RNAi for that gene. So I'm going to talk about the corazonan lineage as a model, and just to give you an example of the types of questions we can ask when you look at one group of cells. So as I mentioned, there's one per hemisegment. They, within 6 hours, undergo cell death, and all the neurites, axons, and synaptic debris are going to be cleared by 12 hours. There are also 3 per brain hemisphere that don't undergo cell death, that are nice internal controls, so you can see that they're fine. And if we start to make single-cell clones with Spaghetti Monster or other tools, you can see they have this really beautiful morphology. Just to our surprise, actually, if you look in every segment, every one of them looks a little different. They're not the same in every segment, but they all undergo cell death. They're all going to get cleared. So why is just this cell type interesting? We started to think about this actually back at UMass when a graduate student named Özge Testimish started to look at these. And what she did was to ask, okay, of the pathways we've been studying, so Draper, which is MAGF10 in the mouse, and this is MBC, CRAC, DSED12, or DOC180 Elmo in a mouse, what role do they play in the clearance of these cells? And so here's a control animal. These are all 18 hours APF, or head aversion. There's a brain there. It's just all the debris has been cleared, okay? So first she knocked out this GEF for RAC, and she found there was all this debris, right? So that's good. There's a role for SED12, okay? What if she knocks out Draper? Well, she sees the opposite phenotype. Now she sees all the dead cell bodies are still there, but the debris is gone, right? And then if she knocks out both, she gets everything, okay? So what does that mean? That means if you just look at this one neuron, somehow this neuron, when it kills itself, tags its cell body for clearance by this pathway, and the rest of it by clearance from some other pathway, right? So even in that one neuron, things get complicated when you look very carefully. And we don't understand exactly how that's tagged, But as you'll see, what we started to do, we're looking for mutants which gave phenotypes like this, additive with Draper, to see if we could find what is that other receptor. Okay, so here's Jan Schick. He's a postdoc in the lab. Everybody says this, but this guy is a monster. He's amazing. He's going on the job market. He's really, really terrific. I am going to fail to tell you how wonderful he is, but I'll try. So Jan Schick comes to the lab, and he had been trained in Arash Bazarula's lab in metamorphosis. He studied cell death in the salivary gland. And so he really took to this project, and neuro was new to him, but he wanted to understand how do these animals, how do they do this phagocytic process as a first step. So here are the astrocytes before they transform. Here they are 6 hours later. He did this old-fashioned trap-seq, which you may know what that is. That's where you take and you express a tagged ribosome, and you pull down RNA from just one cell type, in this case astrocytes, at different time points. And he got a really beautiful expression data set of a whole bunch of genes that were upregulated. He picked through those for the ones he thought might be most interesting. And then what he did was a screen where he just got RNAis, one or more than one for each gene, drove those in glia, and then he had made a corazonin Lex A to drive GFP. So he's tagging that with GFP, he's doing the screen in glia. And how many do you think he got? That is not a lie. He came to my office almost in tears one day. And so you might wonder, why do I put this up here? It's because we are forever being shown expression data sets, and people make conclusions about what cells are doing based on that. You can't do that, okay? Those genes are probably important, but you can't say they're important until you actually go test them, right? And the other thing, so that's one, is like, we're fly people, we test things, right? We should go in there and break it and see what happens. And the second is that the people in the lab will tell you, I moan all the time that flies are a very sophisticated genetic system, but if you actually look, how many people last year did a screen where they tried to screen for things that enhanced a phenotype? The answer is probably 0, right? And we can't do that. Biology is more complicated than that. Worm people are starting to do that a lot more now, but if you look at a yeast paper, they start with like 5 mutations, and then they do their genetic screens, and then they find whole new pathways, okay? So I'll get off my soapbox now, but I think that we need to think about that as a field. So as he was crying, I said, no problem, just go back and do it in a Draper background. And so Rachel De La Torre, because I don't have to do all the work, right? It's easy. So Rachel worked with him to repeat, and it's another point, right? To do that screen, you have to do the cross, grow the animals up, wait for a white prepupa, right, which is a 1-hour window, and pick it, and then time it for 12 or however many hours. And he has slept many times in the lab on the floor, so he's a very dedicated postdoc. So they did that screen again in a background where they also knocked down Draper, and they got, this time, 6 hits. Not a huge number, right, but that's better than nothing. And now one last thing I'll say is many people might be thinking, okay, but it's RNAi, it's not a knockout. And that is true, but RNAi generally works really, really well. I would say maybe 20% won't work based on our experience with all the pathways we studied. For the most part, I think if you really could knock out that other 100 and get a phenotype, we should have seen it. Okay, so one of the genes we found was a molecule called borderless. So here are the corazonin neurons again. This is at 12 hours after preparing information. Here's Draper again, you get some debris. Here's what borderless looked like if you knock it out on its own, and then if you do the double RNAi, what you see, and make a note. You might be thinking, why aren't the cell bodies there? It's because it's just astrocyte, and the previous one I showed you was an all glia. Okay, so Draper, weak phenotype, but something. Borderless, not really anything. Together, additive. Okay, it gets a bit more convincing when this is after pruning. So here's controls. Here's the Draper knockout. You start to see the cell bodies again. Here's borderless, nothing. And then when you look at the 2, you start to see additivity. Okay, so again, not a smashing phenotype, but there and consistent and real. Okay. If you look at the expression of a borderless reporter where there's GFP integrated into the locus, it's off. These are the astrocytes labeled with GAT. By 6 hours APF, it's on at very high levels in those phagocytic astrocytes. It encodes a IGSF molecule, which has been shown by others to play an important role in neuron glia signaling through a co-receptor or a receptor called turtle that's in neurons. So borderless is in the glia, turtle's in the neurons, and in the visual system it's important for wrapping of those neurons. Okay. So Rachel got a turtle cherry, a tagged version of turtle, and looked, And this is just to show you that the cherry signal does not overlap with the astrocytes. They look like they're in largely non-overlapping domains. Borderless is all over the astrocytes, as I showed you. So that's consistent with them being different places. And one thing that was really interesting is if you look at the expression of this reporter, turtle cherry, this is the edge of the neuropil, right? And you can see the neuropil really nicely. But what you don't see is cell bodies out here expressing turtle, right? And if we look at that a little more carefully and we label just the corazonin cell bodies, and then we do some cross-sections, it looks as if turtle is excluded from the cell body and really accumulates in the synaptic neuropil. So the model is that it's expressed in this neuron, but somehow it really gets tethered out here in the synaptic neuropil. Based on that imperfect marker, and we're looking with other things. So that's interesting. Turtle is expressed in the corazonin neurons. So here's a ventral corazonin neuron in the ventral nerve cord. It is not in these DL corazonin neurons, those three that don't die. So obviously, an interesting question is if we put it there, do they die? The first answer seems to be no, but we're still working on that. Does it do anything interesting to the neurons as they die if you knock it out? So here, get rid of that so you don't look at it. These are the cell bodies in here. We're staining with DCP1, which is a cleaved version of a caspase, which is one of the things that gets turned on as these cells start to die. So here's a couple cell bodies. You can see purple. And if you look at the neurites up above the ventral nerve cord as they project into the brain, you can start to see co-localization with that cleaved DCP. If you just knock down turtle in the corazonin neurons, first you see that those neurons start to not activate cleaved caspase, and the neurites also tend to not show cleaved caspase. And this is at 2 hours when a lot is happening. And if we look at 6 hours, to our surprise, what we found is that knocking it out just in those corazonin neurons rescued the vast majority of them from cell death, and most of their neurites appeared intact. Okay, so there's we think, IGSF molecule out in those neurites that somehow signals back to the cell body and says, now's the time to die, right? That's exactly the kind of thing we'd like to try to figure out at a molecular level. If you let it go further, so to 12 hours, that doesn't save all of them, but here's a curious feature, right? You now see 2 live cell bodies and some neurites, and if you look from animal to animal, it's always the same segment where you find those 2, right? And I'll just tell you that in other experiments, we found other IGSF molecules which allow other neurons and other segments to exhibit this behavior. So we're now thinking that layered over that whole nervous system may be a code, like some of the ones we've heard about tonight, of IGSF molecules that are used initially to construct that nervous system, but then also used later in a spatial way to help deconstruct it too. Now, we got a lot of work to do that, but that's the sort of model we're going forward with. And remember, we found this neuronal molecule based on that enhancer screen with Draper, even though borderless itself had a weak phenotype. So there's a lot of questions left. How does turtle actually activate degeneration? How does it signal back to the cell body? Those are kind of redundant. But the cell biology of that signaling event is very interesting for a number of reasons. We initially were thinking borderless as a receptor might be, a recognition receptor for debris and that it might work with Draper for recognition. But now we're thinking maybe actually what it is a trigger for remodeling, and the enhancement we see is sort of the additive effect of not eating quickly and just triggering it a little late. That's our model. We need to do a bit more, but there are other reasons we don't think borderless is probably a phagocytic receptor. But integrating those two is going to be an important question for the future. So that was one project that spun out of what Yun Shik had started, where he mentored Rachel, who was initially a postbac student and is now a grad student in the lab. But before we even did that RNA-seq, he got very ambitious, and he just wanted to ask, on a broad scale, what molecules are required for these glia to transform and phagocytose the corazonin neurons. And so he worked with Amanda Jefferson, a research associate in the lab. Rachel also helped, and Amy Sheehan, who's a senior research associate and lab manager, to do a pretty ambitious screen where they did essentially the screen I told you before, where we had glial-driven RNAis, and we just labeled the corazonin neurons with GFP. And then we collected, basically, as far as we could tell, every transmembrane secreted or signaling molecule encoded in the fly genome, multiple RNAs for many of these. and then repeated the whole screen, right? And that was a lot of work. That was 3 or 4 people doing about 6 months worth of work. And we were looking for anything, right? Anything where when you knock it out in the astrocyte, you get a good phenotype. Now, one of those, there's a lot of things that can go wrong, right? And one of the categories of mutants we got were ones where the astrocytes actually never grew right in the first place, so astrocyte developmental genes. And that spun out into a project on the TREE1 receptor, which is most famous for driving migration of germ cells, and that's on bioRxiv now. But it's also required for astrocyte growth. So that was sort of a byproduct that another postdoc in the lab, Jakun Chen, who's now moving to UNC, just had published. But the results of the screen from the glial astrocyte phagocytosis of the corazona neurons were really good. So these are the different phenotypic categories. There were some where there was actually no death of the neurons at all, and this was myoglianin, which is a TGF-beta that was first discovered by Takeshi Iwasaki with Tsumin Lee. He found 124 genes he could knock out where he got some combination of cell bodies and neurite debris, and then a couple of others where they looked like the Draper phenotype, and a couple where we just got the neurite debris. Now, this we put up for a reason. Anyone who knows what that is, this is trehalase, right? It chops up a sugar. We have no idea why RNAi to trehalase would lead to a blockade of cell corpse engulfment, but we're taking ideas. This is the fun of being in a fly rite. You get really weird phenotypes that might not be expected. But Yunshik chose to focus on tweak, and I'm going to tell you a bit more about that now. And there's a few reasons. So first, it was one of the strongest phenotypes we got where we had lots of debris left. So here's a control animal. Here's tweak at 12 hours. If we did our genetic homework and got mutants, we got very similar phenotypes. So here's a null and then a null over deletion. Interestingly, it was not really strongly required for the removal of the cell bodies. I'm not showing that in this experiment. If you go into the adult and ask, is it required in other contexts for glia to eat axonal debris, so we would cut axons, let them fall apart in the olfactory system, they do it just fine. So tweak, it seems to only be really crucial in this context. So what is it? It was interesting to us because it's this really beautifully conserved molecule. So from yeast all the way to humans, it's got about 40% amino acid identity. It's enormous. It's 565 kilodaltons, right? So think of cloning. That's an 18K bcDNA that you got to put back in a fly. That's not easy. It's among the top 20 or 30, we think, biggest proteins in the fly. Despite that, it had no known functional domains. It had no known functions. But it was expressed in mouse and human astrocytes. So I thought, okay, we should be able to figure out what this is. In addition, there are a number of human disease mutations which are highlighted there that cause really severe brain malformations and neurological disease. So there's reason to believe it's interesting from the perspective of human disease. So Yunshik combed the literature, and if you look really carefully and do a screen of supplementary material, which is a good thing to do, you'll find it was found in a screen in C. elegans for failure to engulf cell corpses. It's called LPD3 in worms. The human version was found in a screen for regulators of phagocytosis, a genome-wide screen with guides for magnetic bead internalization. And it was only really required for the big beads, not the small beads. Interesting. It's also required for internalization of Legionella. And Legionella actually enters the cell in a sort of, the cell tries to phagocytose it and it escapes phagocytosis and it infects. Okay, so that all makes sense that in other animals it's doing phagocytic things. We looked at a GAL4 insertion as a reporter, and as we might think, it is not on in L3, and then it gets really nicely expressed just in the astrocyte. This is a nuclear reporter, that's why it looks like this. Really beautifully at these early phases of metamorphosis. We can do things like say, all right, well, do the glial cells know they should be eating, and have they activated this phagocytic program? So here's control, and in a single cell clone, and this is 2 hours into metamorphosis where we're extaining for Draper. So all that red is activation of Draper. So the cells are starting to phagocytose. And if you look in tweak RNAi or mutants, you can see they still upregulate Draper. They begin to start to eat things. And then it looks like they just fail. So they do have phagocytosis a little bit, and they acidify vesicles, but they really fail to sort of gear up and do it at a high throughput rate like wild type. So if you look at single-cell clones, this was the most striking phenotype. Here's the nucleus of a single astrocyte. Here it is at L3. Early after transformation, they have these beautiful vesicles. Those just don't seem to form in tweak mutants. And I won't belabor it, but there's a ton early in wild type. They don't form in tweak, and you can rescue that by re-expressing it back in astrocytes. So we're faced with this dilemma of still not knowing what this molecule did or what phase of phagocytosis it blocked, but we thought it was early. So Schick went and collected, phagocytosis is obviously a very well-studied phenomenon. There's great markers for each of the steps along the way. He got UAS lines or other types of markers and developed each one of these so he could really beautifully ask where along the way does a tweak mutant block this. Here, for example, is a marker of early phagosomes. This is what it looks like in the larva. You start to see these little circles forming. Those are early phagosomes about 2 hours in. Later, those start to fuse and form those bigger clusters, which you can see with markers for late phagosomes. And it's a little easier to see if I put them both together. And when he looked at tweak, he basically found you never even get to this early phagosome stage, these nice little vesicles. Okay, so it's frozen very, very early in the process. He then went and said, okay, well, what about PHEGFP? That's the earliest marker for nascent phagosomes, so just as they're coming in. And if you just look at controls, you see nice PHEGFP in L3. It's maintained for a couple of hours, and then as the phagosomes mature, it fades in wild type. And it just is not sustained, and it goes off in a tweak mutant. Okay, so it looks like we're really freezing at this very earliest phase of phagocytosis. And we were still stuck with this question, right? Like, what is tweak? We chopped it up. We tried to put it back. Nothing worked. And what saved us, actually, was structural biology. So out came AlphaFold, which I'm sure many of you have heard of, this thing that can predict structures really beautifully. And within that data set was the human version of tweak. And it's too big. It could only do a third of it. But if you looked at that, it's this interesting molecule which has these really large beta sheet structures, a single transmembrane domain. And if you rotate it, it's a big tube. And if you look at where the hydrophobic residues are, they're all on the inside of that tube. So we worked with Isabel Bacongas at the Volum to reconstruct full-length tweak with AlphaFold, and that's what it looks like. And I can say that Schick has gone on with Sarah Clark and Eric Guo's lab to basically solve the structure to, I think, 2.6 angstroms now, and this is really what it looks like. So AlphaFold was right. And what it looks like is a huge tube with a single transmembrane domain that has a very hydrophobic pore, right? So what's that doing? Well, it turns out there's this new family that has been discovered, and primarily this work was pioneered by Pietro de Camilli at Yale, where these are bridge-like lipid transfer proteins. So they are large molecules that essentially link the plasma membrane, which is throughout the cell, to different things like mitochondria or Golgi or lysosomes. And they are essentially fire hoses for lipid transfer, and they just pour lipids from the ER to that next destination, it's believed. Now, a good example is ATG2. Here's what ATG2 looks like. It's got this nice hydrophobic cavity. What it does is link the growing membrane of an autophagosome to the ER, and that allows that expansion membrane to grow, right, and expand and become big and then do its digestive events. So I'm sure you can all guess where we're going with our model now. Our model is that at this initiation of phagocytosis, these astrocytes have to go from non-phagocytic to massively phagocytic, upregulate tweak. That allows them to transfer a huge amount of lipids so that they can do the phagocytic event and form these little vesicles. Okay. Now, if we look with markers, this is called D-mapper. Here's a third instar larva. You can see it's sort of there in punctate, but by 2 hours into metamorphosis, you see really beautiful ERPM contacts, which is the marker for, at the surface of the cell. And in the tweak, and in the tweak animal, you just don't get those, okay? So it seems like it's a really important structural component of these ERPM contacts, and it's crucial for the astrocytes to march down that phagocytic path. And finally, he engineered into the genome a couple of these human disease mutations, and they give phenotypes just like the null mutant. So we think the human disease mutations are probably loss of function too, at least the ones we tested. So to wrap up this, I'll just tell you, we have a lot of important questions, but it seems like it really does form this tubular structure now that they've gone so far with the cryo-EM. In cryo-EM, actually, is pretty amazing. They took a small portion of their sample, they did mass spec on it, and they found 3 at least 2 beautiful binding partners that stick to tweak, and Yunchik's already knocked out a couple and seen similar phenotypes. And we think they may be transmembrane domains that anchor that other part of tweak that don't have a transmembrane domain. Is it a bona fide lipid transfer protein? We wanted to work with Pietro to answer that question. It turns out that you have to pay like $40,000 per purification, so I'm going to let Yunchik answer that when he goes to his own lab. And stay tuned, or we'll wait for the technology to get better. It's definitely at ERPM contact sites. It seems to stabilize them, and the human disease mutations are loss of function. So, that was a really unexpected direction we took from doing a genetic screen, but that's sort of the fun of the fly, right? We started realizing that even this one cell, the corazonin cell, is complicated in how it gets eaten. It has sophisticated signaling events to drive signaling from the neurite back to the cell body to drive death, and then the astrocytes do some pretty amazing things cell biologically to eat them, okay? And now I'm going to finish in just a few slides to say, okay, that's one group of cells. What if we do more, right? The fly is really an untapped reservoir for interesting questions. And, standing on the shoulders of a lot of work that led to a lot of the connectivity data we saw today and the subsets, this whole data set of Gal4 lines is available online. You can look through them. Amy went through and looked at every single one of those 7,000 for ones that were expressed in nice subsets of neurons and asked which ones are strong in a nice subset. Let's order those. We got 200. We looked at those during remodeling, and we found about 17 different groups of cells that show really interesting new pruning events. Okay, that was Amy and Yunchik. And now we're just trying to get our heads around, okay, if you look elsewhere, what do we see? What's the same? What's different? So, for example, here's one which, if you look, it's whole cell elimination. Here's a pair that's up in the brain where you prune this really big axodendritic field. If you do things, after you define the cellular type of pruning, if you do things like knock out the ecdysone receptor, caspases, et cetera, with really easy tools, you can start to ask which ones do what. And the point I'll make here is just that this is caspase-dependent ECR-mediated death, but this is not, right? This doesn't require caspases. It doesn't require ECR. That's interesting because every other cell type we've studied and others have studied requires ECR as that timing signal. So if we take that group of cells and start to do screens for suppression, we should find new molecular pathways. Now, the final thing I'll show you is that Katie picked one line. This is the student who was formerly in Avi's lab. She's leaning over to tell Andy that. She worked on this, right? And this always amazes people who work in mice. But you got this complicated pattern. Okay, you watch it remodel. You can see it gets very clean. Okay, in this pattern, there's 2 cells. There's we're calling the beat, and we're gonna have to rename it. I didn't realize there were so many beats. Whoever gave the talk earlier, it's beat X. We'll call it for now. Lateral or medial. Has a very complicated pattern. It undergoes pruning. The lateral cells die. The medial cells remodel. And what's interesting is that the remodeling of those medial cells still happens even if you block ECR. So that was a big criteria for us because we wanted to find new things. So in a very sort of DOLAB-like approach, we identified different segments. We looked at how they underwent cell death. There's really 2 cells. Here's the beat lateral cell. It's very kind of boring and just undergoes cell death. Here's the medial cell, which has this beautiful field that projects up into the brain. And this is not super interesting. It undergoes cell death, but it does so in a segmentally specific way. So segments 5 through 7 undergo cell death. That made us think of Hox genes, and this is for people who don't work on fly. You all know this is conserved. You'd be surprised at how many people don't. We could talk about that. It will be a never-ending battle we fight. But abdominal B is in these segments. If you knock it out, you start to get those ones that die to live. And if you overexpress it, you can kill them all. Okay, so that can explain the segment-specific elimination of the cells that undergo cell death. And, that's sort of just more patterning. The more interesting part is this medial one, where you get this really beautiful local pruning. All those neurites go away, and you get a single axon shaft back that projects up into the brain. Now, that is ECR independent. It's caspase independent, so it's new. And what's most interesting, actually, is I've been talking to you so much about this astrocyte transformation, how they go from these really nice, sort of friendly synaptic cells to ones that are highly phagocytic. The thing that's really interesting about this group of cells is, so here it is in L3, Here's after pruning. If you just block that by driving an ECR dominant negative cell autonomously, then you can block most of the pruning that takes place in that neuron. Okay, so this is somehow getting a signal from that astrocyte as it transforms to undergo pruning. And that's exactly the kind of cell type we really want to study in detail. I can tell you it's not myoglianin. That was an obvious guess. It must be something else. So I'm going to stop there and just sort of reiterate that this is a really sophisticated process that happens. A lot of interesting cell biology happens. We know very little about the molecular things going on. We're going to try and use the fly to understand that and sort of across the nervous system. There are new types of remodeling that go on out there, and that should lead us hopefully to new pathways. Flies, I don't have to convince you of this, are a great model to pick those things apart and to study astrocyte biology because I'm confident they're more like any of the other glial subtypes if you were to compare a fly astrocyte to a mouse astrocyte than some of the other cell types. That doesn't mean the other ones aren't useful, but this is really a great model, I think. And we can use these to find genes that are important, some of which are already being studied in the context of neuronal remodeling in mammals. So I will stop there. Let me just point to Yun-Shik, who was really the leader of all this work, Rachel, who did the borderless work, Katie, who did the work on the beat neurons, Lede, Amanda, Amy, and several others helped with the expression analysis and surface proteomes. Thank you for your attention. Sorry I went over. Let's go to the bar and talk more. Thank you. Yeah.
We have some time for questions. I'm going to start with a question because I have the mic.
Okay.
So that was.
You don't want to start with a trainee?
Oh yeah, I do. Okay.
Mentoring.
Trainee has a question. Raise your hand, please.
Or just go while you're walking up there.
All right, got one. Wait.
Sorry, Andy.
Hello?
I was just
Can I ask?
hanging out there.
So
Yeah.
I had a quick question. One was if you could provide lipids in a tweak null background, would you see phagocytes formation? And the other one was if you could express something that had a redundant function in a tweak background that had a function similar to tweak, would that also rescue the formation of phagocytes or phagocytic structures?
We have attempted to use dietary lipids to rescue the tweak phenotype, and all I can tell you is that Yun-Shik thought it might work a little bit. The problem is, of course, is this all happens once they go in the pupal case, so they stopped eating, right? And so you have to either get them in there before they enter it or inject or something. So that's a maybe. If we express something redundant, would it work? As far as we know, tweak is the only bridge-like lipid transfer molecule that links the ER to the plasma membrane. So if we overexpress others, it might do it by an indirect supply, but we wouldn't know which one to add to reconstitute that link. We honestly just haven't done those experiments, so we don't know.
Yeah, thank you. Great work. It's definitely nice to see how much effort people put in there. So, yeah, one thing. So you said that either the somer or the neurons itself are tagged. One question would be like, can you think about like how they tag themselves and how they do make difference between the somer and the neurons, so different compartments? And the second was like, compared to the abdominal B labeling, like, could you think about why those in the posterior are seem to be prone, to get erased?
So, why are they differentially tagged, and how do the ventral nerve cord ones die, but not the ones in the brain?
Yes, and also the difference between soma and neurites.
Yeah, so the difference between soma and neurites, we don't know. Everyone thinks of corpses as just corpses, right? They die and they are all tagged the same way, and that's not true. In fact, if you take a corpse and you kill it and drop it on a microglial cell, it can eat it right away, but an astrocyte can't. If you take that corpse and let it sit in a dish for 8 hours and then you dump it on both can eat it. So a corpse changes over time. It ages, the signals on its surface change. So we don't know if it's a timing issue where the surface changes and then it becomes a viable phagocytic target, or if, for instance, the cortex glia, which are the ones that probably eat the cell bodies, they definitely have Draper, and so do the astrocytes. So, like, why the difference? We don't know. The assumption is that the neurites must tag themselves in a way that engages a receptor we haven't identified yet. And that was one of the things that we were hoping to find, and may have in that screen that Yunshik did, where we found the corpses went away just fine, because we did it by knocking out all the glia, but the debris stayed. So that's a sort of to be continued. There's no obvious candidate yet, but that's how we're thinking of it, and that's how we're going to approach it. And then the idea would be to find the ligands for those receptors.
And differences in PS or staining or something? Did you see something there?
Oh, phosphatidylserine?
Yes.
Yeah, that's a tough one. It's almost certainly there. Any cell that falls apart exposes it. There's a million things that can bind phosphatidylserine. I'm not convinced anyone has really shown me that that's the so-called eat me cue. There's a good chance. But there are a lot of, there was one up there called Seymour that is a co-receptor for Draper that may bind phosphatidylserine. There are several bridging molecules, like C1q, for example, can bind phosphatidylserine. In the fly, Eric Barrica's group has shown that MCR, macrocomplement related, is probably playing that role upstream of Draper. So if you forced me to put a bet down, I would say MCR binds phosphatidylserine and then binds Draper and allows it to eat things. But why that doesn't allow it to eat the neurite debris, I don't know. It's a really good question, yeah.
So I have a question. Who decides which neuron should be eliminated? Is it like the neuron tells the glia that, oh, okay, I'm now healthy, so it sends out signals, recruit the glia to kill them? Or do you think the glia has a way to decide which neuron should be eliminated?
My simple model that seems to make sense with a lot of the data ranging from the Purkinje neuron work that I showed you through synaptic pruning is that either a synapse of a number that need to be trimmed or a whole cell I believe the neurons are making the decisions, right? They fire, they figure out who should stay, who should go. And then the weaker ones, if it's a synapse or the ones that's going to die, send out a signal that basically says, I am competent to be engulfed. And then a glial cell will come along and say, all right, I'm here, execute your program and I'll eat you. And then they do, and then the glia clean it up. So it's a conversation that requires glial cells in some ways we don't understand yet. And that's also true in the worm, if you look back at the old, really nice engulfment studies. But how exactly it happens, I couldn't tell you. I think all I would claim is there's good evidence that the engulfing cell helps push the synapse or the whole cell over the edge. And the challenge is to figure out how, what is that molecular signaling event. In DLGN and RGCs, Beth Stevens would probably say, well, maybe C1q sticks to the ones that are weak, and then that's how the microglia or astrocytes recognize it, right? But that's the million-dollar question. We don't know. Yeah.
Hi, Marc. This is Jim from Quant's Lab. I talked to you probably a couple of years ago.
Mm-hmm.
Yeah. So today I have a question about the tweak. So it's a lipid transport protein.
Mm-hmm.
So do you have any idea what type of lipid it transport? Because there's so many different types, like sterols and phospholipids and glycerols.
We have no idea. Pietro is working very hard to figure that out. And we told him, he came to visit and we talked to him, and he has done some lipid transfer with one of the smaller versions that was possible. When we got the cryo-EM work going and were able to pull down enough to do cryo-EM, we called him up, and that's where I said, I'm not paying $40,000 for a prep, right? That's because you don't need a ton for cryo-EM or for mass spec, but to do those biochemical assays, you need a pretty strong amount. And so those questions are hard to answer and won't be answered until this technology gets better. But it would be really interesting if you could regulate which lipids go through, if you could modify it in some way to change the profile of lipids that went through, but we just don't know, right? This is a biophysicist problem. Maybe you could ask Yunshik in 10 years and he'll answer it.
So, hi.
Yeah.
Hi, Marc. Beautiful talk and very informative. So, I have questions in two parts basically. So, one is direct, that does this tweak have some role in adult brain, and the second is we know that neuron-glia interaction is very important to eliminate protein accumulates or some extracellular debris as well. So in that case, can we use this tweak as a tool to kind of forcefully eliminate debris which is accumulated in the cells?
Are you still talking about tweak, or are you just talking about
tweak?
Yeah. This answer to your second question, I have no idea. I would struggle to come up with a way to do that. And then the first question, is tweak required elsewhere? We think tweak is required in all cells. There's a pretty healthy maternal contribution. The nulls are not great, you really have to work to get them to L3 or pupal stages. We think that it's, we've also done some work, for example, with Shengzhu in worm, because in worms what you can do is we had this idea that maybe what tweak is responding to Draper localizing to the target, and then Draper recruits it so that it can deliver lipids to the right place, right? That was one model. So we looked in worms where you can actually watch a corpse get engulfed in really fine detail in live imaging. And getting rid of tweak had no effect on SED1 localization to the corpse or vice versa, as far as we could tell. So, how you would target it somewhere, I don't know. We think it's probably expressed in all cells. All cells use it. If you knock it out, they may be able to deliver lipids in another way, either with vesicles or something, but at a much lower level. And we think that that astrocyte transformation, where you take the morphology and so radically transform it, and then just have to engulf all this material, basically all the synapses and a lot of the axonal and synaptic debris in the neuropil, that flies have evolved tweak as a way to upregulate in astrocytes to give them enough lipid to do that job, because it's the one place that it has been seen to be so dramatically upregulated, according, for example, to Pietro, right? So he was very excited. So that's all I can really say in response to that. Avi.
So is this the same giant molecule that Patrick Verstricken showed counteracted PIP2 levels at the NMJ? So isn't PIP2 kind of a strong candidate? Have you tried looking in synaptogenic mutants to see if it suppresses some of the phagocytic defects?
We have not done that. PIP2 was altered. I showed the marker for that. And so, that was the result they saw, right? Is that if you look at the NMJs in a tweak mutant, you have less PIP2. Mm-hmm. Whether it transports PIP2, we don't know, right?
It was a confusing paper, and I think there were a lot of tools missing because it was a giant protein, and it was 2003, and there wasn't a way to deal with it. But I wonder.
There still isn't.
You don't think of presynaptic terminals as being actively phagocytic, so why was tweak particularly important there?
Well,
or did they not get the cell autonomy of that?
I would not think of it as a protein, as a phagocytic protein. That's what we're studying. I would think of it as a protein that can deliver lipids to the membrane if the membrane needs to do a lot with vesicles, like synaptic vesicles, right? And so maybe in that context, it's just delivering the lipids that are needed. It's believed there are ways to deliver small amounts of lipid, but when you really need bulk transfer, then this family gets engaged. And when it comes to ERPM transfer, it's tweak. So, you're welcome to plenty of mutants and tools if you want to look at the NMJ. But we haven't, yeah. And that is the origin. Actually, some of the tools Hugo was nice enough to share.
Hi, over here.
Yes?
Are there different isoforms that have different lengths that might affect the length or the rate at which it delivers lipids?
I can't remember. I'm sorry, yeah. We looked at that at one point, and I think the answer is yes, but mostly change the very ends of it, not... I'd have to look. I don't want to be completely wrong. But that's an interesting question, yeah. Let him go drink beer.
Yeah. Let's thank Dr. Freeman for a really nice talk.